Today, the day will rather start with a lot of lectures, so we will have this lecture now here. The next two lectures will both take place downstairs in the big lecture hall where you always have coffee. This will be the lecture before and after lunch, and only in the afternoon we will then come back to this room and we will again start working a lot with Skyline, with first a Sky Jam from Brand and then some more tutorials for you to do. Okay, that's the plan for today. And yesterday you have already heard a lot about how to design a targeted proteomic assay, and then how this data looks like and how you can confidently identify your peptides and proteins of interest from such targeted proteomic data. But we hardly touched on how we really quantify peptides and proteins from the data. And this is what we will do now in this first lecture here this morning. So I would like to have a look with you at the basic principles, or some basic principles of quantitative targeted proteomics. And in particular, I would like to discuss with you the differences between relative and absolute protein quantification. And I've structured this lecture into 4 parts. So first we will look at this very basic question of how and why mass spectrometry in proteomics can at all be quantitative. Then we will look at the principles of relative protein quantification, followed by absolute protein quantification, and if time allows, we will also look in the last part at some specific challenges that are of specific relevance when you do absolute protein quantification. And again, whenever there are questions from your side, just interrupt me at any time and ask. Good. Let's start with the first part, and with the very first, or the first question that you can ask when you think about quantitative proteomics is the question of why are we at all aiming always for quantitative proteomic data, or why is it often not sufficient to just qualitatively describe which proteins are present in a sample and which not. And one analogy that you can use to nicely demonstrate these differences between quantitative data and qualitative data is the analogy to photography. So in a way, you can understand a qualitative description of a proteomic sample like a black and white picture, where in this black and white picture, every pixel only is allowed to be either black or white, to be there or not there, depending on the threshold that you define. And from such a purely black and white picture, you can already get some information. So in this little example, you can see this picture has something to do with greetings from, but still there are many parts of this picture that are blurry or unclear, or you cannot make a lot of sense out of it. While in the moment when you go from this black and white world into a quantitative world, so if you add quantitative information to this picture, in a sense that you allow every pixel to be not only black or white, but to have different shades of gray, you increase the knowledge of this picture, and the true meaning of this picture becomes suddenly clear. So you can, from this picture, get much more knowledge than from this picture, just by adding this quantitative additional information. And exactly this same concept is also true in the world of proteomics. So if you just qualitatively compare 2 sets of samples, like a disease and a control set of sample, you maybe find exactly the same proteins in these samples. So from the pure protein list, you cannot differentiate the 2 states. But when you have quantitative information, you can actually maybe find proteins that are high in control and low in disease, and the other way around. So from quantitative data, you actually can differentiate these 2. Okay, so that's the reason why quantitative data is so much better than qualitative data. Now, the question is, why did mass spectrometry not started quantitative right from the beginning? Why did we only start in the, let's say, the last 2 decades to retrieve quantitative information from our mass spec data? And the answer to that is that there is actually a poor correlation between the amount of a peptide that we have in the sample and the signal intensity that we measure for this peptide in a single MS spectrum. So the situation is here once more explained based on one example. So if we look at this protein, and if we digest a specific protein into its peptides, then in theory all these peptides are equimolar, as they all originate from the same starting amount of protein. But when we measure these peptides in a mass spectrometer with any type of mass spectrometric approach, we will always see such a whole range of different mass spectrometric intensities. And the reason for that is that this intensity that we measure here in the mass spectrometer is unfortunately not only dependent on the abundance of this peptide in the sample, but it's also dependent on a whole lot of other factors. Like, for example, the intensity we see here also depends on the digestion efficiency of a specific peptide. So how complete is it cleaved out of the mother protein? It also depends on the recovery of this peptide during sample processing. Maybe some peptides you lose to attachment to surfaces, or they are just not soluble. It also depends on the ionization efficiency of the peptide, how well ionize them during electron spray ionization. It depends on matrix effects. So what's the sample background where these peptides are measured, and does the background enhance or suppress the ionization of a specific peptide? And it just depends on the specific MS detection properties of the different peptides of the different amino acid sequences we're looking at. And it's really just the sum of all these factors together that define what you will finally see here, and this is what we call then always the MS response factor, so the intensity value. And that's why this gets so complicated. However, what people have figured out is, quite quickly, if you just focus onto a single peptide, the intensity of a single peptide, and when you follow the intensity of the single peptide over a chromatographic retention time. So if you measure a single peptide several times with several mass spectrometric spectra over a chromatographic retention time, then you can extract these chromatographic traces, these chromatographic peaks. You have already seen a lot yesterday. And actually this chromatographic peak, so the behavior of the peptide over time, this peak or the area of that peak or the height of that peak, this linearly scales with the abundance of the peptide in the sample. And this is shown here for one example. So this is a synthetic peptide, a so-called aqua peptide that carries a C-terminal heavy-labeled lysine residue, which was purified so that you knew the concentration of that peptide, and then I performed a dilution series experiment of that peptide ranging from 50 femtomole in the sample down to 50 attomole, and you can see that the mass spectrometric intensity that gets measured for this peptide at this different concentration nicely linearly scales. So this means that, for example, if you increase the concentration by a factor of 2, you also increase the intensity that you measure by a factor of 2. And it's exactly this linearity range here that is based on all the quantitation results, or that's the basis for all the quantitation that we do in mass spectrometry. And for such a very nice and beautiful example as I show here, this now seems very logic and sounds very easy and straightforward. However, there's a problem with this range of linearity. And the problem is that in reality, this range of linearity only holds true for a specific concentration range and for a specific mass spectrometric intensity range. And whenever you are above this range of linearity, you run into issues with detector saturation here at the upper end. And at the lower end of this dynamic range, you run into issues with limit of detection, because at one point you will just start measuring background and no longer your analyte of interest, and then also this sample will flatten out at the bottom and at the top. And the two endpoints of this range of linearity is what is called the lower limit of quantification and the upper limit of quantification. What's very important now to understand about this figure is that for any type of protein quantification, accurate protein quantification that you want to do, it is actually absolutely crucial that the data points that you use for your quantitative analysis are coming from this range of linearity. Only then the underlying assumption of our quantification, namely that there is a linear dependency between our measured intensities, only when the points are coming from this range, this assumption will be fulfilled. And in the moment when just one of these 2 points shifts out of this range of linearity, you will get an erroneous quantitative result. And what's also very important to keep in mind is that this holds true for any type of mass spectrometry that we're doing. So it doesn't matter whether these 2 points are coming from label-free analysis, so if they were measured in 2 different samples using label-free mass spectrometry, or whether these 2 points are coming from an isotope-labeled analysis when you do quantification with synthetic peptides and the heavy point shifts out of this range, or the light point. It doesn't matter which type of mass spectrometry you're working with. It's always the same concept. And one other factor that now complicates the whole situation even more is the fact that this range of linearity is not just constant in your measurement, but it's actually something that is peptide as well as mass spectrometer dependent. So every peptide can have a quite different range of linearity. And again, this range of linearity can change if you go from one mass spectrometer to another. So in theory, the only way to figure out the range of linearity for your peptides of interest is to really measure this range of linearity. And this can only be done when you have a purified reference molecule for your analytes. And then you can perform a dilution series experiment and then exactly figure out this range of linearity and where it flattens out and what's the linear equation for your peptide. And of course, working with these reference molecules, highly purified, this is very expensive, and this is why the vast majority of quantitative proteomics data haven't figured out this range of linearity, but what people typically do instead is they just assume that all their data comes from this linear dynamic range. This is what's typically done in the field, and I think it's very important that you are aware of this issue and that for highly accurate protein quantification, in theory, you would always first need to check that the underlying assumption is actually really valid in your measurements. Okay? So now we know in theory how protein quantification and proteomics works and why it works. And if you now want to perform your first own quantitative proteomic measurement, the situation that you face looks like this. So there's a whole range, a multitude of different flavors and different approaches that you can apply when you want to do quantitative proteomics. And the big question everybody has always is which of these multitude of different options is the right option for me and for my project. And of course, it's beyond the scope of this lecture here to go into all these details of these different projects and what's the disadvantages and advantages. But there are a few questions that you can ask yourself and that will define which is the appropriate technique for a given project. And these questions include, for example, to what type of mass spectrometer or mass spectrometric platform will you have access to, as this will define the mass spectrometry that you can do. Another question is how precise and accurate do your quantitative results have to be? So for your project, will it be absolutely crucial to have coefficients of variance between your replicates below 10%? You need to be very, very precise and accurate. Then you will probably have to work with stable isotope labeling. Or is it a project where you are rather interested in very large protein abundance changes and maybe you can live with a 30% coefficient of variance and then label-free quantification is also a good option for you. What type of sample are you working with? So not every type of these different approaches is applicable to any sample type. How large is your project? How many samples do you want to compare? What's your budget? How much instrument time do you have? Or how much budget do you have for sample preparation? And then finally, is also a question, what type of quantitative data do you really need? Do you need relative or absolute data? And as all these first points here are rather project location and person specific, for the rest of this presentation, I would like to focus onto these differences between relative and absolute protein data. Okay. So to bring everybody to the same page, what's actually the definition of absolute and relative protein quantification? So here you see the definition of relative quantification. If you want to perform a relative quantitative analysis in proteomics, you need to work with at least 2 samples. You can have more, but you need to have at least 2. And the output that you want to generate is always a protein ratio. So what you want to say in the end is whether a certain protein is up or down or not regulated between these 2 samples. In absolute protein quantification, the situation is different. You can also work with just a single sample if you do absolute protein quantification. You cannot work with a single sample if you do relative quantification. And the output that you aim for is always a real protein concentration unit. For example, you want to say I have X number of copies, protein copies per cell, or I have a certain amount in femtomole per microgram of tissue extract or per milliliter of body fluid. These are all absolute protein concentration units. So one major difference between the 2 approaches is that in relative quantitative proteomics, you always compare the same protein between samples. That's what you do. In absolute protein quantification, you can do the same. You can also compare the absolute concentration of always the same protein between your samples, but you can do one more thing. You can also compare the concentration of different proteins within the same sample. And that's something you cannot do with relative data. So if this is true, if we have this nice additional feature that we have with absolute data, why are we not all just forgetting about relative quantification and just do absolute quantification in the future? And actually this would not be a very good idea, because the analytical challenges that you need to face to do absolute protein quantifications are much higher, and we will come back to this later in the talk. So it is much more easy to get erroneous results or to get wrong results with absolute data. And this is why absolute quantification should really just be applied to projects where it makes sense to generate absolute data. Okay, so if we come back to this picture of all the different quantitative mass spectrometric approaches, all these techniques can actually be categorized into projects useful for relative and for absolute quantification. And actually, for relative quantification, all the approaches that I've shown here are useful. So with any type of mass spectrometric technique, you can do relative quantification. While absolute protein quantification or the estimation of absolute abundances, you can only do with a handful of those approaches. Okay, and so this brings me to the second part of the presentation, and where I would like to look with you a bit into the principle of how relative protein quantification works. And as we already said, so all of the relative quantification approaches can be categorized into two groups. Some of them are working label-free, and others are working based on an isotope-labeled standard, so they are label-based. And in order to make this relative quantification really clear and easy, I would like to look with you at one specific example. So let's assume we're interested in this target peptide, and we want to quantify this peptide with SRM label-free. What do we need to do? What have you learned yesterday? So what's the first thing that we need to do if you want to quantify this peptide label-free with SRM? Start with a method. So what does it mean? We need a method. So first thing we need is an SRM assay. Exactly. How do I get this? You can have a synthetic peptide and generate it. You can predict it. Exactly. What else can you do? Identify it by yourself or in the literature. Find an MS2 spectra where this peptide was identified. And when you have the MS2 spectra, what do you do? What do you select for this peptide? The transitions, the most intense transitions. Exactly. Oh, maybe I forgot. So the aim is to measure this peptide, of course, in two samples. So in the control and in the disease sample, and to do label-free quantification. Exactly. Now, as we said, we need this SRM assay. What does the SRM assay all include? Which information? So once more, what are the different columns? The what? The IRT time, if you are very experienced and you've done a good job, exactly. You will have IRT information in there. What else? Q1, Q3. Collision energy you must define, exactly. Dwell time, super. And then there was one last thing that you can store as well, but it's not required for before the measurement. Which information do we also store in an SRM assay, but not necessarily before the measurement? The relative intensities of all the fragment ions, exactly. Okay, so this I have now generated. This is the SRM assay that I generated for this peptide. So we will target these 5 transitions. These are all the parameters you just very perfectly named. And then this is the result. So I have measured now this peptide with these 5 transitions in a control and in a disease sample. What do I do next for label-free relative quantification? What do I do with this now? Yeah? Mm-hmm. Exactly. I have many peak areas. How do I have the sum of all transitions? Exactly. This would happen. What would Skyline? This is how Skyline does it. Exactly. To determine the ratio between the two, Skyline would sum up all the transitions of the control sample, sum up all the transitions of the disease, and then it would take the ratio between the two. The advantage when you take this sum of all the transitions is that you weight these different transitions depending on the intensity. So a very intense transition will have a bigger weight on the final ratio than a very low intense transition. However, there are also other ways to determine a ratio for a peptide ratio based on several transitions. You can, for example, also take the ratio of every individual transition and make the average of that. And like this, you weight every transition in the same way. Just that you're aware of it. There are different options. There is no wrong or right, I would say. The way how Skyline does it, it sums transitions and makes the ratio based on that. And in this little example, we would then find that our peptide in the disease condition is 1.3-fold downregulated. And that would be our label-free result, yeah? Well, I mean, if those transitions are coming from a true target peptide that is present, I mean, they should be all there or not, but you're completely right. You can have different interferences when you work with different backgrounds, two different samples, whereas a different background, there might be, I mean, these are now two very beautiful peaks, so I would say they are not interfered, and I'm pretty confident that this is my peptide. But you're right, there are many issues that can happen with this label-free quantification. One issue, for example, also that we don't address at all is here we are measuring a peptide concentration, but very often we want to derive protein ratios in the end. So how do you combine different peptides to a final protein ratio is one issue. Second issue is normalization. So just by looking at this peak, I'm not sure, do I have actually really loaded the same amount of proteins in control and disease? I cannot judge this by just looking at this one peak. So there are many issues actually. Then there is the issue of statistical significance. Is this fold change that I find now here, is this significant? Is it reproducible, or is this maybe not? Is it just within my biological variability? So all these are points that are very important to consider, but I don't want to discuss them here, as there will be a whole lecture, and I think it will be several lectures actually upcoming that just focus on this. So there's Olga Witek coming, who's an expert in statistical analysis of targeted proteomics data, and she will go much more into these things with you on Wednesday and on Thursday, I think. But the basic principle, this was for me important, would work like this. This is how you would label-free quantify a protein with a peptide within SRM. And I would just quickly like to summarize here once more the advantages of label-free relative quantification. So the big advantage of working label-free, it's cost and work effective, at least in the context of sample preparation. So you do not need to spend money on specific labels or further pre-process your sample. It's applicable to all proteins that you could identify and quantify. So you can really do proteome-wide analysis with such type of analysis. You do not increase the sample complexity, so depending on the stable isotope labeling approach that you will work with, for example, if you do SILAC labeling, you generate a completely labeled proteome, and by mixing them, you increase the complexity of your sample by a factor of 2, which can significantly decrease your sensitivity. And that's also one advantage when you work label-free, you don't have this increased complexity. And it's applicable to a large number of samples. However, the major disadvantage, and we touched on that already, is that any variation that happens during sample preparation or chromatography or mass spectrometric performance will immediately impact your quantitative outcome if you do label-free quantification. So for accurate label-free quantification, you really must be especially careful in all these steps and must make sure that you're working consistent and reproducible for all the samples that you want to relatively compare. And that's also the reason why quantitative precision and accuracy in label-free analysis is typically lower than what you can achieve with isotope labeling. Okay, and that also brings me to the isotope labeling approaches. So how does relative isotope labeling work? So just as a reminder, the most important facts about isotope labeling. So what you do in isotope labeling is you generate a reference molecule, a heavy reference molecule that carries one or several isotope labeled atoms. The important concept here is that these heavy references are chemically identical to the endogenous light target, and therefore they have exactly the same behavior when it comes to sample preparation biases. So if you lose peptide, you will lose your heavy peptide in the same way as your reference peptide. They have the same chromatography, the same ionization efficiency, the same fragmentation. So by using these heavy references, you can account for variation that happens from the moment on when you have spiked these references into your sample. And that's why you are more precise and accurate with these methods. And the typical used isotope-labeled atoms are 13C, 15N, and 18O. Also deuterium is used, but it's a bit less favorable because it can induce chromatographic elution time differences. And this is why people prefer to work with these molecules where you have also exactly same chromatographic elution. So all the label-based approaches that I showed you on this overview slide can be categorized into 4 groups. The first group is based on metabolic labeling. So here you introduce isotope-labeled atoms by growing your cells on an isotope-labeled isotope-labeled media. So the typical representative techniques for metabolic labeling is, for example, SILAC or 15N labeling. Then there is the second approach where you do isotope tagging by chemical reactions. So what you do here is you exploit the reactivity of the amino acid side chains or the N or C terminus of your peptides. And the typical techniques used here are demethyl labeling or ICAT, or the 2 isobaric tagging approaches, TMT or ITRAC. Then there is the third group where you introduce your isotopes via enzyme reactions. So for example, you can do your digestion in 18O labeled water, and then all your peptides will carry a C-terminally labeled 18O. And the last group of stable isotope labeled approaches are the spike-in standards. So in this approach, you don't do your labeling globally on the whole proteome, but you generate isotope-labeled references per peptide or protein. So one approach here is the aqua approach, where you synthesize peptides that carry typically a C-terminally labeled lysine or arginine residue. Then there is the Q-concat approach. Have you heard about this before? Does anybody know what the Q-concat is? It's an isotope-labeled protein, yes, but what's special about this protein? Exactly. What you do is you just concatenate into a plasmid all the peptides that are of interest for you. And like this, you generate a new artificial protein that is made up only of the peptides that are of interest for you. And then you overexpress this artificial protein in an isotope-labeled media. You purify it. And then you digest this protein together with your sample of interest. And like that, you produce, during the digestion, a lot of peptides that are of interest for you, all in the same concentration. That's the theory of it. So these Q-concats are also spike-in standards, or then these you can also, in exactly the same way I've just explained how you generate Q-concats, you can also generate a completely labeled protein standard. So don't use just peptide sequences, but use the whole protein sequence and produce an isotope-labeled protein standard. Okay, so let's again have a look at our little example here. And now let's ask the question, what do I need to do now if I want to quantify my peptide here in the control and in the C sample using label-based quantification with SRM? What do I need to do now compared to what we have done with label-free? Yeah? To the transition list. Perfect. That would be one way to do it. The way I have done it here, I was lucky enough. I got a synthetic peptide even. Okay, so here we can even work. It's absolutely correct. One option would be to label the complete one condition and then put them together. Another option is to spike in a reference molecule. And the important point to also know is that if you want to do label-based relative quantification, you do not need to know the concentration that you spike in of the heavy reference that you spike in. The only thing that matters is that you should spike in the same between the two conditions that you want to compare. And then you said it correctly. So if I now go to the mass spectrometer, what's important, I have to add now also the transitions of all of this heavy peptide version to my transition list. Otherwise, I cannot target it with SRM. So now here you see the transitions of both of those peptide forms, light and heavy. And what you can see is that, for example, here we have Y ions and 2 B ions, and the B ions, ion masses are actually exactly the same between the two molecules. Does anybody know why this is the case? Does that make sense? Is that an error? Is it correct? Why? Exactly, the label, this is the heavy label lysine here, so all the fragments that are coming from the B N-terminal end, all the B ions will not carry the label. That's correct. Okay, but isn't that a big problem now for my measurement? I mean, now I will measure the exactly same masses here. Now I will get my one peptide will pollute the other. I will get wrong intensity for these B ions, no? Exactly. Because we have this different Q1 and we have this around 1 Dalton resolution, precursor isolation window that we can achieve in the quadrupole, we are far beyond this 1 Dalton, so the instrument should have no problem to differentiate these two because of the different Q1 mass. Make sense? Everybody got this? Okay. Okay, super, yeah, and that's it. Now we can do label-based relative quantification. So we will measure in the two conditions always the light as well as the heavy, the light as well as the heavy. And from this we can then generate the two ratios here within the sample. And as we have spiked in the exactly same amount of heavy in both of these conditions, we know that the blue curve here is exactly the same, and we can simply calculate the ratio between the two samples by taking the ratio of the ratios. So 2 divided by 1.37, so the peptide is 1.5-fold upregulated in the disease state. Okay, so to summarize the advantages of label-based relative quantification, it's less prone to preparative and analytical variabilities, and therefore label-based quantification is more precise and accurate than label-free. And as you have for all of your analytes always a light and a heavy channel, you are also more confident in your peak identification when you have both of these things, especially when you work with synthetic reference peptides, where you know what's inside, you're even more confident here. But there are also disadvantages. So stable isotope labeling is work and cost intensive. Not all the methods are applicable to every sample type. Silac labeling is not applicable to tissue samples, for example. And depending on the labeling method, a significant increase in sample complexity can reduce your sensitivity. Okay, are there any more questions to relative protein quantification? No, super. Then we will go on and talk about absolute quantification. So I already told you in the beginning that absolute quantification is more challenging than relative, and you should only apply it to a project where it really makes sense to apply it and to go through this pain of absolute quantification. Which are these projects where that makes sense? So here's an overview of projects where absolute data matter. One typical field of application for absolute quantification are protein complex stoichiometries, and we have heard quite a bit about this yesterday at the Science Slam, so quite some of you are interested in protein complex stoichiometries, because what you need to do if you want to compare, or if you want to figure out the stoichiometry, how different protein components of a complex assemble, you need to compare the abundance of these different components to each other in one sample. That's exactly what absolute quantification can do and relative not. Another typical field of application is mathematical modeling. So to determine rate constances, kinetic fluxes, you need to have as an input to solve these mathematical equation absolute protein concentration data. Also, if you want to look at how much energy the cell spends on producing certain proteins, you need to know the protein copy number of these proteins in the cell. Then there is the field of pharmaceutical and biomedical industry. So for various applications in industry like food safety, quality control, sports drug testing, absolute proteins get measured. Also for protein biomarker development, it has been shown that it's clinically relevant to not just know the relative abundance of a biomarker in the sample, but you really want to know in a specific patient what's the concentration of the biomarker in plasma or in urine. And then there is this last field of data cross-comparison. So relative data is usually always relative to a certain reference. And if projects have been generated with different references, it's impossible to compare them. While absolute data can always be immediately compared. And this sometimes makes interesting comparison possible. For example, if you want to compare different experiments that were not designed to be comparable, or if you want to compare data from different labs, different mass spectrometric platforms, or even different organisms, this is often just possible with absolute data, unless you have planned it right from the beginning with relative data as well. Okay, so also when we now look again at techniques that we have in mass spectrometry to absolutely quantify proteins, these techniques can again be categorized into absolute label-based and absolute label-free techniques. And the gold standard in absolute protein quantification is definitely to work with this absolute label-based technique. And what you need to do here is you need to work with a reference molecule from which you precisely know the concentration in the sample. That's the only difference to relative quantification. When you want to do absolute quantification, you need to know the amount that you have spiked into your sample of interest precisely. And when we look at these different techniques that I showed you before that can be used to incorporate isotope labels into molecules, only this last part here is the suitable type of technique for absolute quantification, because only here you can generate highly purified molecules from which you can determine the concentration and then spike them in with a known concentration. Okay, so it's these three different types of molecules that typically are used in absolute quantification, and each of these again has of course advantages and disadvantages. So the major advantage of the aquapeptides, and that's also the reason why the vast majority of people work with them, is their easy availability. So there are several companies out that provide, where you can order these aquapeptides and then they get delivered to you usually within 4 weeks. Also for Q-concates, there are commercial companies available, and a fast way to Q-concates are these ready-to-use kits. So these companies offer, for example, ready-made Q-concates with peptides for all kinases of a specific organism, or other functionally related proteins that might be of interest to customers. However, when you want to generate your own customized Q-concat with specifically your peptides of interest, this will take several months until it can be delivered to you. And the protein standards are finally typically generated in-house. So usually when people work with them, they produce them themselves, which is also, of course, time-consuming and expensive. In terms of price, all of them are pretty bad. So every type of this absolute quantification is expensive. So one aquapeptide is in the range of at least 300 euros per peptide. And then we have learned yesterday you should always quantify proteins with several peptides per protein. So if you need to order 3 or 4 or 5 peptides per protein, it gets really, really expensive. But the same is also true for these other two approaches are also very expensive. Multiplexing, so how many peptides and proteins can you target with these different approaches? So the most papers that are published that use aquapeptides are maybe in the range of 10 to 30 proteins that they manage, simply because they are limited by this high costs. That's the main limitation factor. With Q-concats, as you can generate up to 50 peptides on one Q-concat, you can maybe target a few more proteins, and with protein standards, usually people just really work with 1 or 2 protein standards. That is the protein of interest for them. And finally, precision and accuracy. So the most precise and therefore also most accurate approach of these 3 molecules are definitely the protein standards. And the reason why the protein standards are more accurate is the fact that only with these molecules you can account for incomplete digestion of your target proteins. Only when you work with a protein standard and you spike it in before digestion, you do not really need to worry about digestion efficiency, because it will your target protein and your reference protein should be digested in a very similar way. And this is not the case with these 2 peptide-based approaches. There you will always have issues with this. But also with the protein standards, there are remaining issues. For example, if you have any type of post-translational modifications occurring on your analyte protein, usually your standard protein that got expressed in another organism or cell-free will not carry any of these PTMs. So this will cause errors or stability and expression issues with these standard proteins can also occur. So it's a pretty hard business of producing the ideal reference molecule for you. I think the ideal reference molecule actually doesn't exist. Okay, and then finally, applicability to PTM. So if you're interested in absolutely quantifying PTMs, then you are more or less just left with working with these aquapeptides, because only with these aquapeptides you can quantitatively introduce quite easily post-translational modifications, at least some post-translational modifications. You can phosphorylate or acetylate your peptide during synthesis. While with these two approaches, it is much harder to quantitatively introduce a PTM on those peptides. So typically what people hear is they just indirectly then look at the non-modified peptides and say then something indirectly about the modified, the PTM carrying peptide. Okay? So then again, let's have a look at this one example. We again back to our peptide, two conditions, and now we want to do absolute quantification of this peptide, and we want to do the most straightforward way of absolute quantification, which is a single point calibration quantification. What does it mean, and what do we need to do now compared to relative label-based quantification? Sorry? Yes, that would be not the single point, but the calibration, multiple point quantification. So one step ahead. Currently, I want to do it the most straightforward way. Exactly. The only difference compared to what I've told you before is that you spike in your reference in a known concentration. So let's assume we spike in this synthetic reference in 50 femtomole per microgram. Then what you can do is, by looking at these ratios that we obtained before, you can simply, based on the ratio, calculate that if the blue curve is 50 femtomole, then the red, the light endogenous analyte, should be 68.5 or 100 femtomole, just based on the ratio. However, to do this type of easy calculation, you apply two assumptions. The first assumption is that you make is that you have carried out your measurements within the linear dynamic range of the mass spectrometer. And the second assumption that you make is that this linear correlation, this linearity that you apply, that you assume, has a slope of 1 and an x-intercept of 0. Only with these two conditions you can make these very easy mathematical equations that a 1.37-fold increase in intensity means a 68.5 femtomole concentration. Okay? And unfortunately, these two assumptions are not always necessarily true or hold true. So an even more accurate way to do absolute quantification is to not rely on this single point analysis, but to perform a multiple point calibration curve. And what you do here is you simply spike your internal standard, so your reference, into your endogenous analyte samples at various concentrations, not just at one point. Your endogenous will always stay constant, that's the background, and the heavy reference you spike in at various concentrations. And like this, you can determine the linear equation, so the linear dependency of your peptide in your measurement. And then you can use this equation to convert your measured endogenous intensity into a real concentration value. And the big advantage when you do it this way is that you can account for linear peptide responses that do not have a slope of 1 and an x-intercept of 0. Yeah? Yeah, I mean, it depends on at the concentration. So for sure, if you will have background, this is exactly if you have high intense background interference. But sometimes, I mean, if your endogenous peptide is high as well. So if you spike in your peptide at a high concentration, you will not necessarily see this, that there is some interferences at the lower end or at one point. So just by looking at the chromatogram, doesn't necessarily mean that you will be able to spot issues when you just work with the single point calibration. It's really the knowledge of the linear behavior that will tell you this. You need to have several data points. I mean, if you see with a single point calibration that it's just a bad peak, that already what you spiked in is very noisy, then you can assume that it will not be a good quantitative result. But that's, well, just general. Just by a single point measurement, you cannot spot it, no. Yeah? Sorry, one. Exactly. That's that's even better to do it that way, if you have this. What was your question?
To come back to the next question, also if the value that you calculate is close to the value of your signal point, can you say that the error is smaller?
Perfect, exactly. That's exactly the reason why you should always aim for spiking in your heavy reference peptide as close as possible to the endogenous intensity. If you have a perfect 1-to-1 ratio, then if you don't have then a slope of 1, intercept of 0 will not affect your result. That's the reason why you get also more accurate results with being close to 1-to-1. But if you quantify proteins that are changing over many samples, you sometimes just cannot do this, and you will have to have also bigger differences. Yeah? Sorry. Areas. Well, but this is the peak area. So this intensity here is what I mean with the peak area. I mean, yeah, you have two different. When you look at the chromatographic peak, you can have two outputs, if you like. You can have the peak height. Maybe this is what you extend as intensity. But when I talk here, I always work with peak area anyway, and I would always work with peak area is usually more precise or let's say more reproducible than peak height. Okay, so yeah. Exactly. So you do not need to do for every peptide an own calibration curve. Of course, you can just spike in all the peptides that you have and get you don't in one dilution series. If I got your question correctly, of course you can make them all together, but you need to do several injections per peptide. That's the big advantage, and that's a disadvantage, and that's what I want to say here. So to do this multiple point calibration requires multiple injections per sample. But you can, of course, do several peptides in one at once all together. All of them that you spiked in together. So the best way, so if you don't want to do such a calibration curve for all your samples of interest, take every sample and spike it and measure it several times with different spiking concentrations. What people typically do is they first generate an external calibration curve once, and based on the equation that they get from there, this is what they use then for their future measurements. Yeah? Yes. I mean, I would definitely do this. First of all, because then you will also, not even any background, but your background, your representative background of interest. Also, then you will find your lower limit of quantification and not just anything. And there actually, in the moment when you work in water and without background, you very often have issues that in the moment when you reduce the concentration, effects like losses to surfaces will get more and more severe. So you will actually get weird behavior just because of surface attachment and stuff like this. I would always try to avoid working with pure low concentrated solutions. Will be very tricky. Okay, so now we have discussed, that was everything I wanted to say to absolute label-based quantification, and the last field that is now missing is just the absolute label-free abundance estimation. And we in the lab use an abbreviation for absolute label-free, which is ALF, and I will use this now also very frequently throughout the rest of the presentation. So ALF means absolute label-free abundance estimation. So in the literature, you find actually publications that did ALF on using all different types of mass spectrometric quantification values. So there are papers that describe these approaches based on spectral counting as an input, precursor ion intensities measured with shotgun proteomics, as well as fragment ion intensities measured with SRM or swath. And there are also a range of different approaches described in the literature, how, based on these different inputs, you now estimate absolute protein abundances. And all these different approaches rely on slightly different assumptions and mathematical equations, and I will come back to this in a minute. But the general difference between these two approaches, label-based absolute quantification and label-free, is that in absolute label-based quantification, you always work with one reference per analyte. You always have for every peptide the corresponding analyte. This is also why it's so expensive. You need to have all these different references. In absolute label-free, you do not have a reference per analyte, but you work with one reference calibration curve per proteome. And I will show you again in 2 slides how this works. So this slide you have already seen, and I've explained to you that we have this issue that even though we have peptides that are more or less equimolar, as they all come from the same protein, still they will have very different response factors in the mass spectrometer. And what all these different ALF approaches that are published try to do is they in some way try to normalize or to correct for these different MS response factors. This is the basic what they all try to do. And they try to do this using different assumptions. So the top N approach, for example, tries to normalize for differences in response factor by simply just taking the few most intense peptides per protein into account for quantification. And the underlying assumption is that every protein of a proteome produces a few very good flying peptides that have the same MS response factor. That's the assumption. And this assumption holds true, better true for some proteins than for others. So for sure there are some proteins where this assumption is very wrong. But looking over a whole proteome, this assumption that every protein has a few peptides which show the same response factor seems to be reasonably okay. This is what papers have shown. The IBAC approach is another approach where you take all the measured peptides into account per protein, you sum up all the intensities that you have measured, and then you normalize this intensity to the protein size. So in case of IBAC, you normalize to the theoretical number of peptides a protein can produce. In the ENSAF approach, you do the same. You also sum up all peptide intensities per protein, and you normalize by the protein length, just another type of normalization. And in APEX, you also sum up all peptide intensities per protein, but you normalize to the observability of the peptides. So in this approach, there is an algorithm included that can predict how likely it is to see certain peptides and not to see certain peptides. And while all of those approaches here usually only work with unique peptides, prototypic peptides, so they simply disregard any shared peptides, this Scampi approach is another statistical approach that was designed to on purpose deal also with these shared peptides. But no matter which of these different approaches you work with, the outcome is always the same. You will end up with a list of proteins, and for each protein you will have one computed intensity value using any of those approaches or equations here. And this intensity value you can already use if you want to relatively compare the abundance of those proteins towards each other. But if the purpose of your analysis is to really determine the concentration of a protein in the sample in copies per cell, then you still need to convert these MS intensities into a real absolute concentration value. And this you can do by working with such a calibration curve. So what you do is you select over all the proteins that you have measured a few anchor proteins. And for those anchor proteins, you need to accurately know the concentration in the sample. And you can do this by using any of the stable isotope labeling approaches, or you can, for example, use spike-in proteins from which you know the concentration. And with this knowledge, you can now make a linear correlation by plotting this known accurate concentration versus the computed label-free intensity. And the equation of this linear correlation you can then use to translate all the measured intensities of all your peptide into absolute protein concentrations. Okay? Is that more or less clear? It's in a way like the IRT concept. It's exactly the same. With IRT, you generate a linear calibration curve to translate one unit into the other. Okay, so what now people usually who work in this field have a major problem with is that which of these different approaches that are now described in the literature shall I use for my project, ABAC, ENSAF, and there are even much more which I didn't list on my slide. And therefore, a colleague of mine, George Rosenberger, wrote an R script to address this issue. That's called ALFQ, and it's also freely available on CRAN. And this R script can import various different proteomic data sets. So you can import spectral counts, but also MS1 intensities from shotgun data or fragment ion intensities. You can import the absolute abundances of your anchor proteins. And then this R script will automatically run those 4 ALF approaches that I introduced to you. And for each approach, it will generate the corresponding calibration curve, and it will estimate the average error based on those anchor proteins for you by doing a Monte Carlo cross-validation analysis. So for each approach, you will get the estimated error, average error, and this average error you can then use to first of all look how these different approaches behave on your data, and to choose which one is the one that you maybe want to work with and that you trust most. So to summarize everything that I said about absolute protein quantification here once more. So the advantages of working with label-based approaches is that they are more precise. So the average CV that you can achieve by using spiking network peptides is often in the range of 10%. It's very good. However, it's expensive, and usually you can just target a few proteins with these approaches. That's the big advantage of using ALF and doing label-free quantification. You can do it proteome-wide, and the average error is in the range. But you need to face the fact that the average error that you can expect from such a type of analysis will be in the range of twofold. This is what people have shown repetitively in various papers. So the value that you will get in the end from this data will be, in average, twofold off from the value that you will get with stable isotope labeling. And with this error in mind, and by coming back to this application scheme that I showed you, I personally think that probably the best technique, if you're interested in protein complexes, or if you're working for industry or clinical application, in these cases you really need to be pretty precise. So a twofold error will be too big for you. And this is where using stable isotope labeling quantification is the method of choice, I guess, what you need to do. But in these other two fields of application where you look more globally onto the proteome, and where it's important to have absolute data for a lot of proteins, and where you can maybe better live with this twofold error, these are typical fields of application where also ALF abundances can be very valuable and be a good method of choice. Are there any more questions to absolute quantification? No? Good. Then I will very quickly run through this last part, and this is really just a collection of some specific tips and tricks for people who would like to do absolute protein quantification, what's important to consider. And many of these things we actually said already. So the first specific challenge that is even more a challenge than it is for relative quantification is the optimal selection of peptides. And that's this slide I already showed you yesterday that describes these quantitative peptides, and where if you plan to do absolute quantification, it really makes a lot of sense to try to stick to these rules and to find peptides that are fully cleaved, that are stably cleaved, that are fast cleaved out of your proteins, to use peptides that are not chemically modified and that do not carry a PTM. And really the best way to address these things is also always to rather work with a peptide more for quantification than with a peptide less. So rather even go for 5 peptides for absolute quantification than to start with only 1 or 2. Then you have no chance to figure out these outlier peptides from your analysis. The second major problem with absolute quantification is the complete protein extraction. So usually what we all do, what is always done when people report protein copy numbers per cell in papers, is the assumption that we have extracted all the proteins to 100% from our sample. But what's actually true is that, of course, we will have global protein losses, for example, due to incomplete cell lysis, some cells which just never lyse. Or you can also have local losses of just specific proteins, for example, membrane proteins or insoluble proteins. You will never get as good in solution as you think, as you want. So ideally, what you would like to do is to identify these losses that are happening, and there's no way to avoid them, and to account for them. But the big problem is that, at least to my knowledge, to date, there is no reliable and accurate technique that you can use to identify the fraction of these losses that are happening here. So the only thing that you can try to do is to keep these losses minimal. And that means that you really, when you plan to do absolute quantification, you need to think about how to have the very best extraction procedure for your sample type. I, for example, worked a lot with yeast and MTB, and we have found that doing several extraction rounds definitely increases the extraction efficiency a lot. So just to take the same cells and extract them over and over again actually yields a lot more protein than when you do it just once. This biocycling technology is maybe a promising new technology that can help with this as well, where you lyse your cells at ultra high pressure, or you can think about using solubilization additives like surfactants or heterotropic reagents or organic solvents. These are all things to consider and which are very important for absolute quantification. Then there is the issue of protein peptide losses during sample handling. So next to the loss that can happen right from the beginning when you extract your cells, there are further losses that can happen on the way during sample processing. And there are 3 types of losses that you can consider. The first type of loss can happen before you actually digest so when you're still on protein level, before you do the digestion. For example, due to protein precipitation, or they just degrade in your lysis buffer, or they attach again to surfaces. And the solution to this type of problem is to work with standard proteins. So if you directly after cell lysis manage to add a standard protein that reflects your native protein, then you can assume that you can account for these losses. But even after digestion, the loss goes on. So you can then, for example, during C18 cleanup, lose peptides. And the solution here is to, again, work with protein or peptide standards. If you have spiked them in before the C18 cleanup, you can account for those losses. And then there's the third type of loss that can happen that is often maybe a bit forgotten, and that's the loss of the synthetic standard molecule. So if you work with these aqua or protein standards, they're actually super nasty. We just said this already. When you have highly pure concentration, highly pure solutions of these things, you can lose peptides while you pipette it from one vial into the other by attachments to surfaces, degradation, freezing, thawing cycles can be a big issue for specific peptides, for others not at all. So the solution here is whenever you work with so low complex and low concentrated standard solutions, try to make sure to work with low binding vessels and low binding pipette tips. So there are special tips that are especially useful for pipetting peptides and to keep these losses minimal. And I have done some tests with them, and it can really make a huge difference for some peptides, for others not at all. Try to work with glassware instead of plastic whenever possible. Use complex sample background or BSA solution to block vessel walls to keep these losses minimal. Aliquot your standard stock solution to avoid freezing thawing cycles. So really when they arrive, aliquot them and always take out the fresh aliquot, and only purchase standard in solution. So there are also some companies who offer them lyophilized, but I would be very suspicious, as you never know how much of this lyophilized powder that you get, you actually manage to get back in solution. Okay, and the last issue I would like to quickly mention, and this we actually mentioned many times already also yesterday, is specific and complete protein digestion. So while for relative quantification, incomplete and unspecific digestion can be tolerated, sometimes it's even desirable because you even increase your protein coverage, right, if you have lots of missed cleavages. For absolute quantification, complete and specific proteolysis is crucial. And what can you do? Well, you can work with these isotope-labeled reference proteins. That's always a nice way out of this digestion issue. But you should also try to maximize your digestion efficiency. So if you plan an absolute quantification project, you need to think a bit about or test a bit your optimal digestion protocol. You can vary time, temperature, buffers, digestion enhancing additives. And one protocol that we here in the lab work a lot with whenever it comes to absolute quantification is a protocol where we first digest our proteins with lyc at 6 molar urea, and then we use trypsin as the second digestion step at 2 molar urea. And this protocol has been shown in this paper to increase digestion efficiency and to be especially valuable therefore for absolute quantification. But I don't want to say that this is the only and best way to do it. It's just something that we have found here in the lab to work very well in our hands. Okay, and that's the last point I wanted to make. So take home messages. it would be great if you all remember from this presentation that protein quantification is based on the assumption that mass spectrometric intensity scales linear with protein abundance. But be aware, this range of linearity is peptide and mass spectrometer dependent. For every project, the optimal quantitative approach must be selected. Unfortunately, there is not one that fits all, and this selection depends on a lot of different questions that you can ask, whether you need relative or absolute data, how precise and accurate your data needs to be, sample type, sample number, budget, and so on and so forth. Accurate protein quantification is definitely more challenging than relative quantification, and absolute typical fields of application for absolute protein quantification are protein complex stoichiometries, mathematical modeling, and then applications in industry, biomarker research, or data cross comparisons. And there are still these specific challenges that are remaining for absolute quantification. We discussed these 4 of them: complete protein extraction, optimal peptide selection, complete digestion, and protein and peptide losses. The definitely major one, in my opinion, is still this complete protein extraction. And as long as this issue is actually not solved, I think there still remains actually a quite big question mark on how accurate our absolute protein quantification results really are. Nobody can really tell, as long as we don't find better tools to address these issues. With this, I'm at the end of the presentation, and I thank you all for your attention, and I'm happy to take any more questions. Yeah? As early as possible. I spike it in usually even before digestion. Yeah. Shall I repeat the question? Okay, so the question is, what's the best time point to spike in a peptide reference, right? A protein reference, by the way, you would anyway spike in before. And I always say the best time point is to spike it in as early as possible. So I spike it in even before digestion. Yeah?
Do you have a trick to avoid the absorption of peptides at low concentration?
Yeah, so when I, for example, do dilution series, and you need to prepare the dilution series. I mean, I always spike the dilution series for the measurement also into a background, but you need to first prepare the dilutions, and then I always work with a low concentration of my sample background already in these dilutions that I make. So I block the even the glass vials of my dilution series with some cellular background. It can be very low. I think I use 0.05 micrograms or so. But I can see a huge difference whether I do this or whether I don't block it. I think I'd always take the same background that I use then later on in high concentration, but I think you can use anything. But it makes also huge difference, and it gets more and more severe the lower the concentrations get. I never tried this to flush it and to remove it again. I always have it in my sample. When you do SRM or these targeted proteomics analysis, I think it's no problem. Especially if you use the same background that you use later on, I would recommend doing this. But I never tried flushing or anything like this. Okay, yeah, we are already quite late, I think, 5 minutes over time. So I'm around for the rest of the week anyway. You can come and ask questions anytime. And now we make coffee break until 9:30. 10, 10:30.